So good day everyone, my name is Milhor Marabalti and today I'll be sharing with you an important topic about the flow of genetic information. And I hope at the end of my video presentation you'll learn something new today. So, first slide. So this image showed the certain direction kung saan nagpo-flow yung ating genetic info from the DNA. So this is the illustration and I hope masundan nyo yung mga meaning nila. The first process is gagawa muna tayo ng sariling copy of the DNA. Then, alam naman natin na ang bawat cell sa katawan natin is identical sa DNA. So that we call the first process DNA replication. So the second process is we make DNA to become an RNA or mRNA, or ang meaning niya is messenger RNA. So that called transcription. After that, from RNA to protein, we call that translation. So next is the cell replication. We should know first the two fundamentals of DNA replication. So the first one is the cell replication. So meron akong tanong sa inyo. Why need to replicate the DNA? Bakit nga ba kailangan ni DNA mag-replicate? So we need to replicate the DNA for order the cell to replicate. So remember that important na mag-replicate si DNA in order for the cell to replicate. So there is certain stages for the cell cycle. We have the G1, S phase, G2, and the mitosis. The first one is the G1, kung saan lumalaki si cell. Cell grows in size and prepares for DNA replication. The next one is the S phase, kung saan si DNA replication, doon nangyayari si DNA replication. Sa G2 naman is the cell prepares for division. Dito rin nangyayari yung final punctuation or the final subphase of interphase at dapat replicated na lahat ng organelles. And then the next one is the cytokinesis and the mitosis, that nagre-result na si mitosis into the two cells, and that's the final phase. So, lagi natin dito tatandaan na sa S phase nangyayari yung DNA replication. So, you need to remember that DNA replication is nangyayari sa S phase. Saan nga nangyayari si DNA replication? Sa S phase. Next slide. So, semi-conservative. So, semi-conservative replication means that each new DNA molecule contains one original parent strand and one newly made strand. So, half of the original DNA is kept and the other half is newly created. So, it means, ang semi-conservative replication daw, it is a way ng pag-copy ni DNA. So, kapag kumokopya si DNA, the two strands separated. After that, yung mga old strand ang nagsisilbing guide para gumawa ng mga new strand. Kaya each new DNA ay may isang old strand at ang isang new strand. Ito ang dahilan kung bakit tinatawag itong semi-conservative. So, I have an example for this. So, this is my example. So I have an example video. So, imagine nyo ha. Okay. So, parang zipper ang DNA. Binubuksan muna ang zipper. So, pag binuksan yung zipper, naghihiwalay ang dalawang strand. Okay, yan. Diba? Pag binuksan si zipper, naghiwalay yung dalawang strand. So, bawat side ng zipper ay ginagawang pattern. Okay ha? Yung bawat side nung zipper, diba? Dalawang side yon. Ginagawan siya ng mga, okay, bawat side ng zipper ay ginagawang pattern para kagawa ng bagong side. So, at the end, may dalawang DNA molecules at bawat isa ay may old and new. May old strand and may new strand. So, sa madaling salita, semi-conservative replication means each new DNA has one strand and one new strand. Okay? Yun ang tatandaan natin. The semi-conservative, meaning niya is, yung mga DNA is, meron siyang one old strand and new strand. Okay, I hope naintindihan niyo yun. Next slide is, ito naman yung stages of DNA replication. The first one is the initiation. So, meron akong ginawa ditong illustration for you to better understand initiation. Okay? Ito siya. Okay. The stage 1 is the initiation. Let's start with the first stage, which is the initiation. So, bago tayo magpatuloy, importante munang tandaan ang direction ng DNA. The two DNA strands are anti-parallel, meaning magkasalungat ang direction nila. So, one strand runs to 5' to 3', while the opposite strand runs to 3' to 5'. Parang two-way street sila. Magkatabi sila pero opposite ang direction. So, ang unang enzyme ay helicase. Sa helicase, ang nagbubukas or nag-a-unzip ng DNA double helix sa origin of replication. Imagine ninyo ang zipper ng jacket. Kapag ninila natin ang zipper, naghihiwalay ang dalawang sides. Ganun din ang ginagawa ng helicase. So, the step 2 is the SSB proteins. Kapag nabuksan na ang DNA, yan, may tendency itong magsara ulit. Kaya pumapasok si SSB proteins or single strand binding proteins. Again, what is the meaning of SSB? So, SSB means is the single strand binding protein. Ang trabaho nila ay hawakan ang dalawang strand para manatiling bukas habang kinokopya. So, the step 3 is the typosomerase. Habang binubuksan ang DNA, umiikot at sumisikip ang strand sa unahan. Kaya may enzyme tayong si topoisomerase. Siya ang nagre-relieve ng twisting and tension para hindi maputol ang DNA. So, the step 4 is the primase. Pagkatapos naman, si primase ang kumikilos. Naglalagay siya ng RNA primer, which is serve as the starting point. Think of it like drawing the first guideline before writing. Diba, hindi makakapagsimula si DNA polymerase kung walang primer. So, kapag handa na ang DNA, maaari nang magsimula ang paggawa ng bagong strand. Yun yung transition natin. Kapag handa na ang DNA, maaari nang magsimula ang paggawa ng bagong strand. So, the next stage is the elongation. So, now let's move to the second stage, which is elongation. So, dito naman ginagawa ang bagong strand. Okay? Sa elongation, ginagawa ang bagong strand. So, this is the illustration. So, DNA polymerase. Ang pangunahing enzyme dito is the DNA polymerase. Again, sino nga yung primary enzyme sa elongation, sa stage ni elongation, is si DNA polymerase. Siya ang nagdadagdag ng free nucleotides upang makabuo ng bagong DNA. Yan, ganyan siya. Ganyan yung process niya. Importante pong tandaan natin na DNA polymerase can only build a new strand from 5' to 3'. Hindi siya kayang gumawa ng 3' to 5'. So, sa side na ang template ay 3 to 5', ang bagong strand ay ginagawa ng 5' to 3' continuously. Ito ang tinatawag na leading strand. Mas mabilis ito dahil tuloy-tuloy ang paggawa. So, the lagging strand dito naman sa my right side is sa kabilang side naman, ang template ay 5' to 3' dahil DNA polymerase only works to 5' to 3'. Hindi niya ito kayang gawin continuously, kaya ginagawa niya ito paisa-isa. So ito yung tinatawag nating lagging strand. So, ano naman yung Okazaki fragments? So, ang Okazaki fragments, sabi ni Sir, ang Okazaki fragments daw ay na-imbento ng isang Japanese. So, kaya Okazaki yung tawag sa kanya. So, ang Okazaki fragments, ito yung maliliit na piraso ng DNA na nabubuo sa lagging strand. Ang tawag dito ay Okazaki fragments. Para silang puzzle pieces. Okay? Para silang puzzle pieces na pagsasamahin maya-maya. So, ano naman yung proofreading? So, the proofreading, habang gumagawa ng bagong DNA, chinecheck din ni DNA polymerase kung may maling nucleotide. Tinatawag itong proofreading. Again, si proofreading, siya yung nag-check. So, ano naman yung proofreading? Habang gumagawa ng bagong DNA, chinecheck din ni DNA polymerase kung may maling nucleotide. So, tinatawag itong proofreading. So, para siyang teacher na chinecheck ang exam bago ipa. Tapos na tayo sa elongation. Let's move on sa termination. After elongation, we now proceed to the last stage, which is the termination. Siya na yung pinaka-huling stage. Okay, this is the illustration of termination. So, the first step is inaalis ang RNA primers. The next step is pagkatapos, pinapalitan ito ni DNA polymerase ng tunay na DNA nucleotides. Yan, ganyan yung process niya. Nandyan sa may mga illustration. So, the step 3 is si DNA ligase ang nagtutuo. So, mahalaga si DNA ligase dahil siya yung nagdudugtong sa lahat ng Okazaki fragment. Parang glue siya na nagdidikit ng puzzle piece. Step 4 is kapag nagtagpo na ang replication forks, okay, tapos na ang pagkopya ng buong DNA. And at the end, at ang final result ay dalawang identical DNA molecules. Each DNA molecule contains one old strand and one newly synthesized strand. Ito ang tinatawag natin semi-conservative replication. So, I hope naintindihan niyo po yung tatlong stages ng DNA replication. Okay, this is our conclusion. So, in summary, the DNA replication has 3 stages. The initiation, where the DNA is opened and prepared. Dito bumubukas si DNA and naghahanda. And then the elongation, where new DNA strands are synthesized. And the termination, where the DNA is completed and the 2 identical DNA molecules are produced. This process ensures that every new cell receives the same complete genetic information. So, I hope naintindihan niyo po yung ating topic sa ating conclusion. Okay. But it matters in everyday life and professional practices. So, in the medical field, understanding DNA replication helps doctors and scientists explain how diseases such as cancer develop. So, cancer cells divide uncontrollably because mistakes can happen during DNA replication. So, this knowledge also helps in developing medicines, improving the genetic testing, and creating treatments for inherited disorders. That's why DNA replication is not just a topic in biology. It is essential for life, healthcare, and modern medicine. So, every time our body grows, heals a wound or replace old cell. So, DNA replication is happening. So, without it, our cell would not receive the correct genetic information. So, kaya dito natin nalalaman na napakahalaga pala ng ginagampanan DNA replication sa ating pang-araw-araw na buhay at sa ating katawan. So, ito naman yung easy mnemonics na ibibigay ko sa inyo for you to better understand or para mas matandaan nyo yung topic natin for today. So, stages. Ito yung 3 stages, ha? Ito, tandaan nyo na yung I eat tacos. So, lagi nyo tatandaan yung first initial, yung I is for initiation, E is for elongation, and T is termination. I eat tacos. I is for initiation, E is for elongation, and T is termination. So, this is the other mnemonics kung para mas lalo ninyo siyang mas matandaan. Okay, this is okay. Remember this, that DNA para mas matandaan nyo yung anti-parallel, di ba? DNA polymerase lovers 5' to 3'. Okay, lover C 5' to 3'. Kahit leading o laging strand, lagi siyang gumagawa ng bagong DNA from 5' to 3'. Thank you. I hope you learned something new today. Bye.